This was a deep water spill, and so I think that the public doesn't readily see the impact. If it didn't wash up on shore, if you didn't see a dead bird today, then things are getting better, right? When in fact it may not be the case at all. It's quite the coincidence that we had a project that had just started in the Gulf when the spill happened.
That was run by a colleague called Chuck Fisher, who's at Pennsylvania State University, and they were studying deepwater corals and cold seep communities. Why they were funded was to actually understand what the unperturbed environment would look like in preparation for that terrible day that might come one day where there was an accident, and you wanted to say, well, you know, what was the impact here?
Few people know that we actually have deep water corals in our deep sea. Principally, I'm trying to understand the life that lives on the corals. We've noticed through our research that there are very intimate relationships with corals and animals live on them. That's of tremendous interest to understand how these corals provide diversity.
My particular role was to address the question to what extent these systems are fueled by the energy that's coming from below, and to what extent they're actually fed by the same process that feed life everywhere else on the deep sea floor, which is detritus raining down from the ocean surface.
Part of our work has been putting out sediment traps. These time-lapse traps that have 20 or so cups on a rotisserie, basically. And every 2 weeks, one cup closes and one opens. It spins around. So it documents all the particles that are raining down. And
what we have right now is a time series that goes from the fall of 2009 until January of 2011. We're going to be able to have data from September to January of 2010 into 11 to know what the 12 months on and 6 months after the oil spill, what that looks like.
Officials from many federal agencies, including the National Oceanic and Atmospheric Administration, the Bureau of Ocean Energy Management, Regulation and Enforcement, and the National Science Foundation, called us to say, you've been out there recently, you know what the baseline observations are for the corals and the animals living with them. Take samples for hydrocarbon analysis, fingerprint analysis, to see if it can be tied back to the Macondo well, 252 oil. And that's what we did in October, November of 2010, and we were 7 miles from the spill. We found this hard bottom area and lots of corals. First thing I noticed was that there's this large red coral called a bubblegum coral, and you could see where flesh, tissue had come off of the coral and exposed the white skeleton. The coral next to it was covered with this brown schmutz-like material that I call it, this flocculent stuff that we subsequently sampled. We stopped the ROV, we stopped everything and said, something's weird happening here, we don't know what. This is as close as we've been to the well, and so let's just stop and treat it as a crime scene. We're going to spend the rest of our dive here and make sure we document this really well, get really important samples for analysis back in the lab, and that's what we did. So we were out there in October of 2010 and went back in December with Alvin. We haven't done enough, and that's why we're going to go back out at least 3 times in the upcoming year to find those coral ecosystems and assess their damage, if there is any.
The decision to use such a large quantity of dispersant and to use dispersant in a sort of previously untested way was potentially of concern, because dispersants, of course, had been used a lot in surface waters, but really hadn't been used in the deep water in that way, certainly not on that scale before.
There was all this talk of dispersants. I mean, they're known to be potentially toxic at some certain level. There wasn't a very sensitive protocol to be able to measure the concentration of these dispersants.
We collect organic matter from the ocean, so we extract it and then analyze it with the idea of trying to describe the complexity and composition of that material with ultra-high resolution mass spectrometry. It's a very powerful tool that has not really been applied to the oil spill research in the past because it's a very recent tool. My sense is that that the public was unsatisfied with the concept that the dispersant was just gone. You know, you could see these big tankers filled with this stuff being dumped in the ocean, and how could it just disappear and nobody know where it went? We developed a method that was 1,000 times more sensitive than the method that the EPA uses for the same molecule. It was just moving out of their detection window. But it stayed in our detection window because we were so much more sensitive. We were able to identify and quantify one of the active ingredients in the dispersant, and that's the first time that's been done for this oil spill.
Liz's study was huge because it mapped where the chemical dispersant was going. By establishing that it was there was a big breakthrough. And then now that that method is available, you know, others could use it to look at, you know, the next set of questions, which is, you know, is this a high enough concentration to be toxic?
There are a lot of other animals that could be affected by the pollution that people don't think about. There's a lot of little grass shrimp and little crustaceans, little mollusks, small fishes, and those animals are really important to the functioning of the salt marsh. We do a lot of work on cnidarians, so those are stinging-celled animals like sea anemones and corals. Nematostella is the example I happen to work on. It's a little kind of maybe 1 centimeter sea anemone that burrows into the sediments of marshes. We got really interested in using this animal as a toxicology model, and in part because the whole genome is sequenced. And so we can study how any or all of the genes respond to chemical pollutants. Before this whole oil spill happened, we had developed a technique. We developed a microarray specifically designed toward measuring responses of Nematostella to chemical stress. So we were really, in a way, well poised when this oil spill happened because we had this microarray ready. We had something sitting there ready to measure responses to chemical pollution in this animal. The major portion of the project has been to do experimental exposures to Nematostella in the laboratory and look at effects on gene expression. And the reason we're doing that is to gain insight into how cnidarians are affected by oil pollution, what sorts of cellular systems are affected, what sorts of cellular and molecular defenses they have. The toxicity of the dispersants like the Corexit, it hasn't been studied yet in a cnidarian. We know very little about how cnidarians respond to these chemicals and also how any estuarine invertebrates respond.
We have an accident, and then everybody says, why can't you marine scientists tell us what's going on? And it's not something that you can find out in a month or several weeks.
And that doesn't happen instantaneously. It takes time and it takes work. That's what science is all about, figuring out the jigsaw puzzle in very, very complicated, interactive, complex systems, such as the ocean.
Our response and our ability to respond to the Deepwater Horizon event demonstrates the value of basic research and the assets associated with that research, which might be vessels, vehicles, laboratory tools, techniques, sampling devices. So that really allowed us to respond in a way that was immediate and was effective.
These are events that are going to happen in the future. There are going to be more of these because the industry is moving further and further offshore, and we will do whatever we can offer to improve the operations, improve the safety, improve the likelihood of this not happening.